What's up? You know what we're going to be talking about today? Biological macromolecules. Carbohydrates, lipids, proteins, and nucleic acids. Stay tuned. Alright, we're just going to go fast with this. So today we're talking about biological macromolecules. Those are carbohydrates, lipids, proteins, and nucleic acids. Those are the things that make up everything in your body. Yeah, more or less, okay? But all of the cells essentially are made up of those four compounds. All of these biological macromolecules that we find all around us, those are not used by living organisms because they're special. They're used because they're available. That's important. Back in the 1950s, I believe it was 1953, I'll have to double check on that. It's this guy will tell you. Actually, it was 1952. There was an experiment that was done. It was called the Miller-Urey experiment. And essentially what they did was they took a sealed cylinder and they supplied a bunch of methane and water vapor and ammonia, hydrogen gas, all the stuff that they actually thought was going to be in the early atmosphere. They supplied all that sort of stuff. They added a bunch of hot water vapor, just they created some steam and added that in there. Then they actually supplied an energy source with electricity. They had this little electric spark, and they let this go completely on its own. And in a very short period of time, what they found were many of these biological macromolecules forming completely on their own. So you take an early atmosphere and give it a little bit of energy, and you get things like carbohydrates forming, and some simple nucleic acids, and some amino acids, and some lipids, and those things form completely on their own. So it's not that living organisms decided to use things like amino acids and carbohydrates because they're somehow special. It's because those are available. We found amino acids on asteroids. These things are not unique, they're common, and that's why life uses them. So with any biological macromolecule, whether it's a carbohydrate, whether it's a nucleic acid, whether it's an amino acid, or whether it's a lipid, we're actually going to have to stick small subunits together. And when we try to stick those two subunits together, we need to free up some chemical bonds in some way to do that. The way that we do that is dehydration synthesis. Say it with me. Dehydration synthesis. In dehydration synthesis, we take water out. So you're going to have an enzyme that's actually going to remove a water, that's going to free up a chemical bond, and it's going to stick two suckers together, whether it's a carbohydrate or whether it's a nucleic acid or an amino acid or a lipid. They all do this. Carbohydrates typically form either 6-carbon or 5-carbon chains, and then those are actually looped together to form 6-carbon or 5-carbon rings. One of the more common ways of classifying whether something is a sugar is if it ends in OSE, it's a sugar. For the most part, carbohydrates are used for short-term energy storage and structure. So the most common ones are things like sucrose and glucose and fructose and lactose. Those are all energy molecules. Cellulose and chitin are structural compounds. The cellulose is found in cell walls of plants. It's what actually gives the trees around me all of their structure. The chitin is what gives insects black ladybug. You ever seen a black ladybug? Lipids are used for a few different things. They're used for longer term energy storage. They're used for structure. They're also hormones. Long-term energy storage is obvious. Those are fats. Things store up fats. There's a lot of calories and stuff in fats. The reason that there's a lot of calories that are stored in fats is that there's a lot of carbon-to-carbon bonds. And the more carbon bonds you pack in something, the more potential energy it actually has. They can also be waterproofing agents. So things like the wax that's on the outside of leaves or the oils that are in mammals' furs to keep them waterproof. All of those are characteristics of lipids that lots of plants and animals take advantage of. As far as the energy form of lipids, we're sort of used to hearing two different terms referring to this, and those are saturated and unsaturated fats. And what it's actually talking about here is a saturated lipid, or an unsaturated lipid, is saturated with hydrogen. So when you have something that has all carbon-to-carbon single bonds, that's a saturated fat. If you have something that has a carbon-to-carbon double bond, that's an unsaturated fat. Now, most of the saturated fats are animal fats. Most unsaturated fats are plant fats, oils, and things like that. We'll also see lipids used in making cell membranes. So one of the main components to the cell membrane is the phospholipid. What we have with a phospholipid are a couple of hydrophobic fatty acid tails. Hydrophobic means that they don't like water, they're afraid of water, attached to a charged hydrophilic phosphate. And if you put those together, then those phospholipids can actually form something called a phospholipid bilayer, where you have the hydrophobic parts of the cell membrane on the inside and the hydrophilic parts pulled to the outside, because the phosphates want to get attracted to the water, the lipids want to be repelled by it, and it forms completely on its own, which is really cool. Something else that lipids will do is form steroids. Now, a lot of steroids, or a lot of hormones, steroid is just another term for hormone, a lot of hormones are lipids. They don't dissolve well in water, but they are able to pass directly across the cell membranes, and that's really important. So what a hormone actually does is it tells a cell to start making a protein. That's what hormones do, is they turn on different genes. So what you need is something that can get through a cell membrane, that can travel through the body, it can get through the cell membrane and get inside the nucleus and turn on certain genes. That's why lipids are so important for it, because the cell membrane is actually made up of lipids. This steroid or hormone can pass directly across it and not get blocked in any way. So things like cholesterol and cortisol and testosterone and estrogen and progesterone and all sorts of different things, adrenaline, those are all hormones that can pass directly into cells. The third biological macromolecule are amino acids. So if carbohydrates and lipids are typically structural and energy molecules, amino acids make up proteins, and proteins do everything. Any kind of chemical process that's actually carried out by a protein. Proteins do it all. So proteins can be structural, they can be hormonal. Things like insulin and glucagon are actually protein hormones. Those activate chemical processes. They don't tell the cell to make a new chemical, they actually just turn on something that's already present. Or they can be enzymatic, and an enzyme is something that carries out a chemical reaction. So the dehydration synthesis that we were talking about just a little while ago, that's carried out by a protein. It's windy. I may wind up filming this whole thing again because of the wind. There are about 20 different amino acids, and while their R group is different, the R group is the part of the amino acid that changes from one to the other, they all have the same background to it. So they all start off with an amino group on one side, a alpha carbon in the middle, and a carboxyl group on the other side. And that's going to allow these things to link up together, because if you look at this right here, you'll see on the left hand side is that amino group. Okay, there's a hydrogen there, and if you look on the carboxyl group, you have a hydroxyl that's sort of sticking right up there. Well, if you've got a hydrogen and you've got a hydroxyl group, then you have the capacity to make water. Remember, dehydration synthesis, we're taking that out. So that's how we can link these things together. Now, what makes all of these different are the R groups. The R groups are different for every single amino acid. I like to think of them kind of like Legos. There's about 20 different major Lego shapes if you don't count like all the funky things like palm trees and stuff. So there's about 20 different shapes of Legos. There's about 20 different amino acids, and you can put those things together in any order that you want and in any number that you want and make virtually any shape that you want. And that's the important thing. In chemistry, shape defines function. Everything in chemistry is reliant on the shape of that. So we go through dehydration synthesis, link amino acids together. When you link different amino acids together, it's actually going to make a three-dimensional structure, and that three-dimensional structure is going to be shape-specific, and it's going to carry out a very, very specific job. Now, sometimes there are mistakes that are made. Occasionally what you'll have is you should have put one amino acid in, and instead you put a different amino acid in, and then therefore you made something that's the wrong shape. Structure defines function. So therefore, if you made a protein that's the wrong shape, it's going to have a different function. It's going to carry out a different job. This is actually what happens with genetic diseases like sickle cell anemia. We got the correct sequence of amino acids that should go together and make normal red blood cells, but if you screw up just one of those, then the shape changes and you wind up with sickle cells. You wind up with red blood cells that don't really work anymore. Okay, so all of those amino acids, they're all linked up together, hopefully in the correct order, and they start to wrap around themselves kind of like this, and they make a three-dimensional structure. And if it's an enzyme, it's going to carry out a specific job, and so it has to be in the right shape to carry out that specific job. Now you can destroy that protein by heating it up or supplying an acid, because what's holding that protein together are hydrogen bonds. And so if you heat it up, that denatures the protein and it comes apart, it stops working. So what would cause us to have the wrong sequence of amino acids? Well, you need to have some kind of information molecule that tells you what the sequence of amino acids are supposed to be. As it turns out, those are nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And the sequence that DNA and RNA go in actually tells us what sequence the amino acids are supposed to do. That's all DNA does. We've been told for a long time that DNA is instructions for how to make everything living. That's not true. It's instructions for how to make proteins. That's it. DNA just tells us how to make proteins. But remember, proteins do everything in the cell. Nucleic acids are basically made up of nucleotides. A nucleotide is a sugar. Depends on if we're talking about DNA or RNA, but if we're talking about DNA, it's deoxyribose. The sugar in RNA is ribose. So we've got sugar, we've got a phosphate, and then we also have a nitrogen base, and there are 4 nitrogen bases. There's adenine, cytosine, guanine, and thiamine. The sequence of those A, T, C's, and G's is what the sequence of DNA is. That's how it can be read. DNA is actually a double-stranded helix, so there's 2 different chains that run counter to one another, and those 2 chains are held together with hydrogen bonds. Hydrogen bonds at this point should sound really important. The DNA stays in the nucleus of the cell. DNA is super, super important. We won't want to do anything to actually damage any of the DNA. So that's always going to stay in the nucleus, but it has to get its information out. Certain enzymes will come in and they will make a copy of DNA in a very specific sequence that's called a gene. That copy is RNA. The RNA travels outside of the nucleus where it's picked up by a ribosome. And ribosomes make protein. So that ribosome is going to grab onto the RNA and it's going to pull it through and it's going to start reading that RNA, and the sequence that the RNA goes in is the sequence that it's going to start attaching amino acids. That leads us to the central dogma of biology. DNA is transcribed into RNA. RNA is translated by a ribosome into a protein. The protein carries out a function in the cell. That's how everything works. Do you really think that that's how everything works? Do you really think that everything is that simple? Does that sound like a lie to children? Of course it is, but for right now, that's going to have to do. See you later, guys. Thanks.
I'm an old man now, can't do nothing. Young folks don't pay me no mind, but in my day I sure was something. For I felt the heavy hand